Hello and welcome to my Edexcel Combined Science Basics presentation for Biology Paper 2. This does not contain everything you need to know to get up to, say, a grade 9. It has had some of the more difficult stuff cut out of it, but it should get you up to at least a grade 6, provided you work hard on it. Do not just listen to this presentation, make sure you're taking notes or making mind maps or drawing diagrams. If you don't understand a bit, pause it, rewind, re-listen to bits, and make sure you're practicing exam questions as well. Let's start with topic 1, key concepts, which is about the structure of cells, including specialized cells, microscopes, enzymes, and cell transport. Cells, the basic structural unit of all living organisms, so all living things are made of cells. You've got animal cells and plant cells, and they both contain these things here. So they both contain cytoplasm, nucleus, ribosome, mitochondria, and cell membrane. And plant cells also contain chloroplasts, vacuole, and a cell wall. So in terms of what some of those things do, we call each one of those parts of a cell an organelle. Okay, the nucleus contains DNA and it controls the cell. The cell membrane controls what enters and leaves the cell. The cytoplasm is where chemical reactions inside the cell happen. Ribosomes make proteins. Mitochondria are where aerobic respiration takes place and that releases energy. And then chloroplasts contain chlorophyll, and that's where photosynthesis happens. Not all cells are the same, so we call the different kinds of animal cell, different kinds of plant cell called specialized cells, and these are cells adapted for a specific role with features that are called adaptations. Okay, two particular ones we're going to focus on. We've got the sperm cell and the egg cell. Now sperm cells, they've got this great big long tail to help them swim. They've got this section here packed with lots of mitochondria to give them energy to swim. They've got this haploid nucleus with only half the normal amount of DNA, so 23 single chromosomes. And they've got an acrosome here to help them digest their way into the egg. The egg, again, it has this haploid nucleus with only half the normal DNA. It's got a thick jelly coat. Okay. And the jelly coat hardens when the first sperm cell penetrates it so that only one sperm cell can enter. And it has lots of mitochondria and lots of nutrients to give it energy to grow. There are lots of other specialized cells, including ciliated cells, red blood cells, nerve cells, xylem cells, and root hair cells. Bacterial cells are slightly different to animal and plant cells, so they've got some common features. So they've still got a cell membrane, they've still got cytoplasm, they've still got ribosomes, albeit slightly smaller ones, and they also have a cell wall like plant cells, but it's got a slightly different structure. They may also have a tail called a flagellum, but not all bacteria have those, and that helps them move. And the big difference is they don't have a nucleus. Instead, they have DNA in two chunks. So they've got this bit here called the chromosomal DNA, which contains most of the genes, and they have these small chunks of DNA like here called plasmids, and they get swapped between bacteria to help spread genes from one to another. We know so much about cells from microscopes. Microscopes produce magnified images, and magnification is how many times bigger an object appears than it really is. Microscopes work using two lenses, the objective lenses, which are mentioned here, and you normally have 3 of those, so there might be times 4, times 10, and times 40 magnification. And we also have the eyepiece lens, which is normally times 10. This gives an overall magnification by multiplying the objective lens by the eyepiece lens. Other important parts of the microscope, we've also got the coarse and fine adjustment dials for focusing it, and we've got the stage where you place the slides. Microscopes produce pictures called micrographs. This here is a micrograph. This one has a magnification of 4,000 times. You need to be able to use a micrograph to find the actual size of parts of a cell, and we're going to use this equation, and it's super easy. If we wanted to find the actual diameter of the nucleus here, we would measure it with a ruler, and let's say that was 20 millimeters. We would just do measured size 20 divided by magnification 4,000, which would equal 0.005 millimeters. We might want to times that by 1,000 to convert it into micrometers. So that would come to 5 micrometers. You need to be comfortable with numbers presented in standard form, which is a way of presenting very large or very small numbers in terms of powers of 10 like this. So something times 10 to the power of something. The power of 10 with a positive power, so here we've got a positive power 6, that is the number of places that appear after the decimal point. So to turn this into a quote-unquote normal number, okay, that 2 is the first number we write, okay, and then you'll notice there are 6 places after that 2. So we go 5, 4, because we've got 5, 4 over here, okay, and then fill the rest of that up with zeros, okay. Numbers smaller than 1 can be represented with negative powers of 10. So again, similar kind of thing, but now the power of 10 represents the position in which the decimal starts. So here, because it's 10 to the minus 6, we're going to have this number here starting on the 6th decimal place. So 1, 2, 3, 4, 5, 6. So the 2 is the 6th decimal place, and then we just finish off the rest of the number here. Your first core practical was to use microscopes. So the first thing is you place your sample on a slide, one of those little oblongs of glass. Next, you stain it with something like iodine to show up the features. You place a little glass square over it called a cover slip, which protects it. Then we place the slide on the stage. This here is the stage. We select our objective lens either 4, 10 or 40 times. We use the rough focus knob to get a rough picture, and then finally we use the fine focus to get a clear, crisp picture. Enzymes are proteins that are found in all of our cells, and they speed up chemical reactions. Enzymes work on chemicals called a substrate, so the substrate gets changed in some way by the enzyme. Now, on every enzyme there is an area called an active site. The active site is part of the enzyme that has the same shape as the substrate. Enzymes work by the lock and key mechanism. So the idea is that the substrate enters the enzyme and it forms an enzyme substrate complex. Whilst it is in the active site, the substrate changes in some way to form the products. And then the products leave the active site, and the active site is left unchanged, ready for another substrate to go in. And this process is repeated many thousands of times every second. One particular type of enzymes is called digestive enzymes, and their job is to break down large food molecules into ones that are small enough to be absorbed into our blood in our intestines. There are 3 main types. First one we have are called lipase enzymes. Their job is to break down fat molecules into glycerol and fatty acids, and that takes place in the small intestine. Then we have protease enzymes. Their job is to break down proteins into amino acids, and that happens both in the stomach with one called pepsin, and in the small intestine with a different one called trypsin. And lastly, we have amylase. Its job is to break down starch into simple sugars, and that first of all starts in our saliva, and then it continues again in our small intestines. All enzymes can be denatured. That means the active site changes shape and the enzyme stops working. So here you can see a normal enzyme with the active site having a shape that matches the substrate. A denatured enzyme like this one, you can see the active site has completely changed shape and it doesn't fit the substrate anymore. This can be caused by high temperatures and very high or very low pHs. We did a core practical to investigate how the pH affects the rate that an enzyme works. The enzyme we used was amylase, and it digests starch solution. Now to do this, what we did was we had a dropping tile, that's this thing here, which has lots of little wells in it, and in each well there is a small amount of iodine. Now the key here is that iodine turns black in the presence of starch. So to set this up, we got one test tube of starch solution and another test tube containing amylase and a pH buffer. The buffer is a solution with a specific pH. And we put both test tubes into a water bath at 40 degrees Celsius to reach the optimum temperature. Then we mixed the test tubes and started a timer. And every 30 seconds, we took a sample out of the test tube and put it into one of our little wells of iodine on the dropping tile. And if it went black, we continued the experiment. And if it didn't go black, if it stayed this sort of orangey brown colour, that meant that all the starch had been used up and we could stop the timer. What we did then was we repeated it with different pH buffers at a range of different pHs. And what you can do is you compare the times for each of the different pHs, and the longer the time, the slower the rate, and vice versa. And what we found was that if we did this, we got a graph like this. So it sort of dips like that. So if that is pH and that is time, we found that there in the middle it had the lowest time, and as you got to a lower pH or a higher pH, the time increased because the rate had decreased. We need to understand how substances enter and leave cells, and in order to do that, we need to understand the concept of concentration. Concentration is the number of particles in a given volume, and we all sort of understand this from our experience of making up Ribena. If we look at these drinks here, we can see that this Ribena has a much lower concentration than this one. If you think about making this first Ribena might have just a tiny amount of Ribena and a whole great load of water, whereas this one might have an awful lot of Ribena in it and very little water. Now, to put that a bit more formally, if we think about actual numbers of particles, if we look at this first diagram here, you can see the red has a lower concentration than the blue because there's fewer reds than blues. If we look at the second diagram, we can see that the red and blue now have equal concentrations because they're at equal numbers of each one. There's 10 of each. If we look at these, this is a little more complicated because here we've got 7 green particles in each of these pistons. But the one on the right here, this is most concentrated because although the number of particles is same, the volume is smaller. And so the overall concentration is greater because it's about the number of particles in a given volume. Now, the first way that substances move in and out of cells is diffusion. Diffusion is the movement of substances from high concentration to low concentration. We describe that as being down a concentration gradient. And you can see that happening in this little animation here. The green particles start off at a high concentration in the bottle and they move away from the bottle towards a low concentration. This is how substances move into the lungs, so how oxygen goes from the lungs to the blood and how carbon dioxide goes from the blood to the lungs. Equally, carbon dioxide enters a leaf from the air where there's a high concentration into the leaf where there's low concentration, and oxygen leaves the leaf by the same process of diffusion. There's a high oxygen concentration in a leaf and it leaves the leaf to the air where there's a lower oxygen concentration. Osmosis is how water moves in and out of cells, and it is the diffusion of water across a partially permeable membrane. A partially permeable membrane, for example, a cell membrane, is a membrane that has small holes in it that allow some substances to pass through, but not others. So in this example, water can pass through, that's the blue particles, but these green particles of sugar cannot pass through. So what happens is water diffuses, in this case, from the left-hand side, where there is a high concentration of water, to the right-hand side, where there is a low concentration of water. How do you know the concentration of water? Well, you've got to think about the amount of solute that is dissolved in it. The more solute, so sugar or salt or something that is dissolved in the water, the lower the water concentration is because there's a load of sugar and other stuff taking up the space. So water moves from where there is a small amount of solute to where there is a large amount of solute, because that is going from where there's a large amount of water to where there is a small amount of water. The final type of transport is called active transport, and this moves substances from low concentration to high concentration. So we describe that as being up a concentration gradient. This is, for example, how minerals from the soil move into a root hair cell. It's also how sucrose moves into a sieve cell in the stem of a plant. You can see an example of active transport happening here. So there are these proteins in the membranes of cells, and what happens is that they grab the substances from the low concentration side and spit them out on the high concentration side. This requires energy, which is why we call it active transport. So cells that do a lot of active transport will have a lot of mitochondria to provide them the energy to do so. The core practical on this was to do with the osmosis in potato chips. So what we did here was we cut potato chips of similar sizes. Okay, we blotted them dry and we recorded the mass. Then we placed them in sucrose. Now sucrose is just sugar, so sugar solutions of different concentrations, and we left them for 15 minutes. Then we blotted them dry and we re-weighed them, and we calculated the percentage change using this formula. Okay, so percentage change equals final minus initial divided by initial times 100. And what we found was that the potato chips that were in a weak sugar solution swelled up, because what happened was there was a higher concentration of water in the sugar solution and a lower concentration of water inside the potato. So by osmosis, water went from the solution into the potato, and the potato cells got bigger, and so the mass increased. In the really strong sugar solution, the concentration of water in the potato chip was higher than it was in the solution. So water left the potato chip by osmosis and entered the solution. So the potato chips in the strong solutions got physically smaller and lost mass. And somewhere in the middle, you have potato chips that didn't change mass because the concentration of water inside and outside the chip was the same. Topic 6, plants. This topic looks at photosynthesis, the core practical photosynthesis, roots, and transpiration and translocation. So the most important job a plant does day in, day out is photosynthesis. Photosynthesis is producing glucose using the energy from light, and the equation for it is as follows. So we have carbon dioxide and water making glucose and oxygen. Now, not on the part of the equation itself, but really important, we need to have light energy, usually from the sun, to drive this process, and chlorophyll to trap the energy in the light in order to make this process happen. This is because glucose is a fuel, it's a store of energy, and the energy it's storing is that energy from the light. Photosynthesis takes place in the chloroplasts inside cells, inside plant cells, because that's where the chlorophyll is. Okay. Now, during the day, photosynthesis is happening as fast as the plant can manage, but the glucose takes up space. So in order to make it take up less space, the glucose that is made during the day gets converted into starch straight away. So the starch is a stored form of glucose made of many glucoses joined together. So if we think of glucose as being sort of little individual circles, we can picture starch as being lots of those circles joined together in great big long chains made of tens of thousands of them, all strung together in one giant necklace. Okay. Now we do that because starch takes up less space than the glucose molecules do. Now, the problem with starch is that it is insoluble, so once it's made it stays where it is because the plant can't easily move it. So at night time, the plant converts the starch into a smaller sugar called sucrose. Now sucrose is just made out of two glucoses joined together like that, and importantly sucrose is soluble, so it can easily be moved around the plant through the phloem, and we'll talk about that later when we talk about translocation. Okay. Now that sucrose goes to all different parts of the plant, and depending on where it goes and exactly what part of the plant it is, different things can happen. So some of the sucrose gets turned back into starch for storage. So if we think about a potato plant, the sucrose that finds itself down in the potatoes under the ground will get turned into starch to store energy for the plant for its future. Some of the sucrose gets turned into glucose. The glucose is used for respiration to release energy for the plants. Remember, all living things respire, including plants. And some of the sucrose is turned into a whole host of other molecules, such as cellulose or proteins or fats and lipids. All these things, these other molecules that the plant needs in order to grow healthily. Okay. Now, photosynthesis takes place inside leaves, and leaves are photosynthesis factories. And this diagram here shows a cross-section through a leaf. And we don't need to know every part of it, but some important parts are the palisade cells here. These are these tall, thin cells, okay? And they are absolutely cram-packed full of chloroplasts. Each one of these little green blobs is a chloroplast. And they've got an awful lot of chloroplasts because this is where most photosynthesis takes place, inside the palisade cell. So those cells need a lot of chloroplasts with all that chlorophyll to enable photosynthesis to take place. Then we have the spongy layer. Now the spongy layer contains all these air gaps. So you can see all these pockets of air here. And that just allows the movement of gases around inside the leaf. So we're going to have gases like oxygen being produced by photosynthesis, and they want to leave the leaf, so they'll travel through those air spaces in that direction. And we've got other gases like carbon dioxide want to get into the leaf, and so they'll travel through the air spaces in that direction. Okay. Now on the bottom of the leaf, we've got hundreds of these little holes here called stoma. The plural of stoma is stomata, so we'll use the word stomata for now. Now the job of stomata is to allow gas exchange. Gas exchange is the movement of carbon dioxide into the leaf and water vapour and oxygen out of the leaf. And the important thing with the stomata is they can open up and close depending on what the plant needs. So during the daytime they'll open up to allow carbon dioxide in and oxygen out, and at the night time they'll close up because it sort of protects the leaf a little bit. Now that happens using these cells. Here you can sort of see some cross sections of them there. They're called guard cells. If we try and draw them, they look a little bit like this. So they're sort of two kind of semi-donut shaped cells like that. Okay. Now during the day these swell up with water, which enters the cells by osmosis, and that changes their shape to the shape you can see there, and it opens the stoma up, allowing carbon dioxide in and oxygen and water vapour out of the leaf. But at night time the water leaves the cells again by osmosis, and then they change their shape, and that closes the stomata up, and that stops gas exchange. But you don't need gas exchange at night because there's no light for photosynthesis. Now, we need to understand the rate that photosynthesis happens at, and when we talk about the rate of photosynthesis, we're going to focus on this idea of limiting factors. So the limiting factor in photosynthesis is whichever factor is present in the least amount, and that limits the rate of photosynthesis. There are three limiting factors we need to know about: carbon dioxide concentration, light intensity, and temperature. And the idea of limiting factors is really one about balance. What it's telling us is there's no point having loads and loads and loads of carbon dioxide if there's not enough light or there's not a high enough temperature. There's no point having super bright light if there's not enough carbon dioxide or the temperature is too low. So whichever one of these things is present in the least amount is called the limiting factor, and that's the one that is holding the photosynthesis back. So it's only by increasing the one that's limiting that you can make photosynthesis happen faster. Now we can show this graphically. So the two graphs for light intensity, that's brightness, and carbon dioxide concentration look very similar. Okay, they've both got these flat sections here. So these are steep sections and flat sections. Now in the steep sections, we say that this is the limiting factor. Okay. So for example, on the light intensity graph, to start with, increasing the light intensity increases the rate of photosynthesis. However, eventually it flattens out and you get the flat section here. Okay. At this point, okay, light in this flat section, light is not the limiting factor. What we're saying is that the plant has more than enough light, and it's the carbon dioxide concentration or the temperature that's holding it back. Whereas in the slope section, the plant doesn't have all the light it needs, and so increasing the light intensity will increase the rate of photosynthesis. Similarly, in the carbon dioxide graph, in this steep section here, the plant does not have enough carbon dioxide, so increasing the level of carbon dioxide will increase the rate of photosynthesis. But again, you get this flat section here, and in this flat section, the plant already has more than enough carbon dioxide. It doesn't have enough light, or the temperature is not right, and so increasing carbon dioxide at this point will not make any further difference. The graph for temperature is slightly different. It does have the sort of sloping section you'd expect for a limiting factor, But then instead of leveling out, you get this nosedive bit here, where it goes down again. What's happening here is something slightly different. This is to do with enzymes, okay, being denatured. So what's happening here in this downward section is that the temperature is increasing so much that the active sites of the enzymes that control photosynthesis are changing shape, and it stops them from working. And at this point, the plant's in real trouble. Now, we did a core practical to investigate the rate of photosynthesis. The aim of it was to investigate or answer the question, how does light intensity affect the rate of photosynthesis? Now, to do this, we used, rather than using plants, we used something called algae balls. Now, algae is this little tiny green sort of single-celled organism that can photosynthesize, and algae balls are just little jelly balls containing lots of algae. Okay. Now, the reason we used algae balls is because you can control them a bit more closely and a bit more carefully than you can with an actual whole plant. So what we did was we got some of these algae balls and we placed them in hydrogen carbonate indicator. Now that's really important because as if you think back to the equation for photosynthesis, as the photosynthesis uses up carbon dioxide, as the reaction goes on, as more photosynthesis happens, you use up more and more of the carbon dioxide. Now that's important because carbon dioxide is acidic. So as you use up the carbon dioxide, okay, you use up the acid, and so the pH of the solution increases. And this hydrogen carbonate indicator changes colour in response to that. It starts out sort of yellowy around pH 7.6, and it goes all the way up to kind of dark blue for pH 9.2. Now you can only get to pH 9.2 if all of your carbon dioxide has gone. So what we did in the experiment was we got 20 algae balls, placed them in some little bottles with some hydrogen carbonate indicator. We wrapped one bottle in foil, that was to completely block out the light so no photosynthesis would take place. Then the other bottles we placed at differing distances from a lamp. So we put one 10 centimeters away, one 20, one 30, one 40, and one 50 centimeters away. And we turned the lamp on, and then we waited for about 30 minutes for photosynthesis to take place. And then we measured and recorded the pH of each bottle by looking at its colour change. And what we found was that the bottles that were nearest the lamp were bluest, had the highest pH, and those that were furthest from the lamp were sort of more orangey and had a lower pH. The ones that were nearest the lamp had photosynthesized much more quickly, so you'd used up all the carbon dioxide and you got a big pH change. The ones that were nearer the lamp, so further from the lamp, photosynthesized more slowly, you used up less carbon dioxide, you got a small pH change. And the one that was wrapped up in black foil didn't change pH at all because there was no photosynthesis taking place. Now, we've looked at the leaves of plants. It's important to understand their roots as well. So the job of roots is to absorb water and mineral nutrients from the soil. But if we look at a root is not just a flat structure. It's coated in these tiny little hairs. You can see each of these hairs sticking out here. Okay? These hairs are made out of cells called root hair cells. And the job of a root hair cell is to stick out into the soil and pull water and nutrients from the soil. Water enters the roots by osmosis. Okay? Nutrients enter the root by active transport. Remember, active transport is when substances move from low concentration to high concentration. That requires energy, so root hair cells have lots of mitochondria to provide that energy. Now, the reason why root hair cells have this long hair on them is just to increase the surface area, because the bigger the surface area, the more quickly the osmosis and active transport can take place. Okay. Now, if we look in more detail at the structure of the root, in the middle, we've got this hollow tube section called xylem. Now, xylem tubes run from the root of a plant all the way up to the leaves, and their job is to carry water and nutrients from the very bottom of the plant, from the soil, all the way to the top, to the leaves where it's needed. But how does the water get into the xylem in the first place? Well, there are two ways. Some of the water will cross from the soil into the root hair cell itself, actually into the body of the cell by osmosis. Okay, and it will then travel from cell to cell to cell, sometimes going across cell membranes by osmosis, sometimes just passing through these little channels, these little gaps between the cells by diffusion. So by a combination of osmosis and diffusion, water moves through the cells until eventually it gets to this area here, which is the xylem. Okay. Some water never enters the cells at all, and it can travel along the cell walls, these brown sections, never actually entering a cell, but just traveling along these cell walls until it gets to the xylem as well. And it travels along the cell walls by diffusion, from high concentration in the soil to low concentration inside the root. And once water is in the plant, we can get the phenomenon of transpiration. Transpiration is the movement of water into the roots of a plant, up the xylem, into their leaves, and then evaporating out into the air. Now this is a really important process because as the water moves up the plant, it also carries nutrients with it, and so those nutrients need to get to the leaves so that the leaves have all the chemicals they need to photosynthesize well. With no transpiration, what would happen is the leaves would sort of essentially kind of get filled up with water because there'd be nowhere else for the water to go. And if the water couldn't go anywhere, there'd be no way for more water to move up from the bottom, and so the supply of nutrients to the leaves would stop. So by evaporating out of the leaves, you constantly make space for more water to come up from the bottom, which creates this continual flow of nutrients from the roots up to the leaves. So it's super, super important. And because transpiration is driven by water evaporating out through the leaves, anything that increases the rate of evaporation will increase the rate of transpiration too. So if it is a windy day, transpiration is faster. If it is a hot day, transpiration is faster. If it is a humid day, transpiration is slower. That's transpiration. Translocation, rather than being the movement of water up the plant, is the movement of sucrose solution around the plant. Remember, sucrose solution is made at night from the starch produced by the leaves, and it's used to transport the products of photosynthesis around the plant. So what happens is the leaves make the sucrose solution. The sucrose solution then goes into the phloem, and phloem is another set of tubes that carry stuff around the plant, and from the phloem the sucrose goes to the rest of the plant. So some of it goes down to the roots, some of it will go up to the flowers, some of it will go to any kind of developing seeds and fruits and things. So it goes all over the place using the phloem. And just in a little bit of detail, if we look at phloem, phloem is made out of two types of cells. We've got sieve cells, which are the actual tube, so the stuff is moving this way through these sieve cells, and we've got companion cells. Okay, there's quite a bit of detail, but the main bit of detail is that the job of the companion cells is to pump the sucrose into these sieve cells, and that keeps the pressure up and forces the sucrose through the sieve cells and spreads the sucrose throughout the whole plant. Topic 7. Coordination, control and homeostasis. This topic focuses on hormones, the menstrual cycle, and insulin and diabetes. Now, a hormone is a chemical messenger that changes the way a part of the body works. Hormones are made by glands called endocrine glands, okay, and they affect organs called target organs. And so an endocrine gland will release a hormone, it will be detected, absorbed by a target organ, and that target organ will start doing something differently. Now, there are lots of endocrine glands. So gentlemen, you have testes, they're producing the hormone testosterone. Ladies, you've got ovaries, they're producing estrogen. We've both got thyroid glands and parathyroid glands producing a range of different hormones. We've got adrenal glands on top of our kidneys, which are making adrenaline. We've got the pancreas making enzymes like insulin and glucagon. In our brains, we've got the hypothalamus here, and we've got the pituitary there, again producing a range of different hormones. So we've got all these different endocrine glands, all producing different hormones that all affect different target organs. Now, on higher tier, if we think about the idea of metabolism, your metabolism is how quickly the body uses the energy in food. Okay, and it is regulated by a chain of different hormones. Thankfully, we don't need to know their names, just initials is fine. So if you've got a high metabolism, that means you use the energy in your food quickly. If you've got a low metabolism, it means you use the energy in your food more slowly. People with a high metabolism tend to be quite skinny. People with a low metabolism tend to be a little less so. Now, the way this is regulated is by something called negative feedback. Okay. Now, a lot of hormones use negative feedback to control the body. So the idea is that if there's a change that increases something, negative feedback will decrease it. If there's a change that decreases something, negative feedback will increase it. And by working in that way, we get a stable environment in the body. So, with the metabolism, the main hormone that controls the metabolic rate is called thyroxine. Okay. Now, thyroxine affects most of the body, and when there's more thyroxine in the blood, the metabolic rate, the speed of your metabolism increases. Okay. Now, it is regulated as follows. The hypothalamus, which is a part of the brain, releases a hormone called TRH. TRH causes the pituitary, which is another part of the brain, another gland in the brain, to release an enzyme called TSH. And finally, TSH causes the thyroid gland to release the thyroxine. Okay. Now here's how the feedback works. If we look at these arrows, the solid ones mean stimulating. So the pituitary is stimulated by TRH, the thyroid gland is stimulated by TSH, and target organs are stimulated by thyroxine. However, the clever bit is that thyroxine, although it stimulates most of the target organs, so most of the rest of the body, it inhibits the hypothalamus and it inhibits the pituitary. So when there is a lot of thyroxine in the blood, the hypothalamus makes less TRH and the pituitary makes less TSH, which means that the thyroid gets less signal to make thyroxine. As thyroxine gets used up, okay, the thyroxine stops inhibiting the hypothalamus, so we get more TRH, and it also stops inhibiting the pituitary, so we get more TSH, and that causes more thyroxine to be produced by the thyroid gland. Now there's more thyroxine, and so that inhibits the release of more TRH and more TSH, and so the amount of thyroxine being produced goes down again. And so you see you've got this cycle where increasing the amount of thyroxine causes a decrease in the hormones that cause it to be made. A decrease in thyroxine causes an increase in the hormones that cause more thyroxine to be made. That's negative feedback. Another higher tier topic, adrenaline. Adrenaline is the fight or flight hormone that its job is to prepare the body for sort of peak performance during times of extreme danger when we either need to run away as fast as we can or fight our way out of danger as sort of strongly as we can. Now, adrenaline is released by our adrenal glands, which are two little glands just above our kidneys, and it causes this fight or flight response, and that's a key phrase. Okay. Now, the fight or flight response involves a few things. Firstly, our heart beats more strongly and more quickly. This pumps a lot more blood around the body, which means our muscles get a lot more oxygen, a lot more glucose gets carried to our muscles as well, and that gives our muscles all the fuel they need to be strong, either for fighting or for running away. Now, there are some changes to our blood vessels as well. The blood vessels to our muscles widen. Again, that increases the blood flow to the muscles so that they get more oxygen and more glucose to give them more fuel to operate. But blood vessels to other organs narrow. So, for example, when you're running away or fighting, you don't need to be wasting any energy digesting food and things like that. So other organs that don't need the blood so much, they get less blood so that there is more to spare for the muscles that are going to be most involved in this fight or flight response. And the last thing is that the adrenaline tells the liver to convert glycogen, which is an energy store, into glucose, which is the fuel our muscles need, again, to work at their optimum rate. So that is the fight or flight response. Heart beats faster and stronger, and blood vessels to muscles widen, to other organs narrow, and the liver converts glycogen to glucose. The menstrual cycle is a roughly 28-day long cycle that women experience from puberty to the menopause, and it's about preparing the body for pregnancy on a more or less monthly basis. So maturing an egg, releasing an egg, and also making sure the uterus is prepared, ready for a potentially fertilized egg or an embryo to be implanted into it so it could develop into a pregnancy. Now, the beginning of the menstrual cycle is defined as when the first blood appears during a period, and we call that period menstruation, and it varies slightly from person to person, month to month, but it's typically from days 1 to 5 is when menstruation takes place. As soon as menstruation is finished, the uterus lining begins to thicken again, and the point of that thick uterus lining is to be able to accept a fertilized egg, an embryo developing, so that it could potentially become a new pregnancy. And that lining thickens between days 6 and 13. Also, during roughly days 1 to 13, one egg in one of the ovaries is maturing, ready to be released, and it's released on day 14 in an event called ovulation. That's when the egg leaves the ovary and starts to travel along the oviduct. The oviducts are these sections here, these long bits at the top corners of the uterus. Okay. Now, on days 15 to 28, the egg makes a slow journey along the oviducts towards the uterus, and if it's lucky, it will be fertilized by a sperm, and if not, it will just come out of the body during the next episode of menstruation. The menstrual cycle is controlled by hormones, mostly estrogen and progesterone, which are the two main female sex hormones. And the last thing to think about on this topic is the idea of contraception. So contraception is about preventing pregnancies, or unwanted pregnancies rather, and there are two main methods. We've got barrier methods, so that's condoms and the diaphragm or cap. Condoms go over the penis and trap sperm so it never gets a chance to enter the vagina and swim through the cervix and into the uterus and meet an egg. The diaphragm sits, this is the cervix here, okay, this is the vagina. The cervix is the sort of narrow gap between the vagina and the uterus, and the diaphragm just sits over the mouth or over the sort of top of the cervix, and it just makes a barrier and it blocks sperm from getting in there. A small number sometimes can make their way around the edge, so the diaphragm is not quite as reliable as condoms, but it's still pretty decent. It's about 90% reliability, something like that. Okay. The main option, particularly the most modern option since sort of the 1960s, has been the contraceptive pill, which uses hormones to prevent the release of an egg during the menstrual cycle. So, menstruation and all that malarkey still happens, but what you don't get is the egg. So you can have unprotected sex without the risk of a pregnancy, or with a much reduced risk of a pregnancy, because the chance of an egg being released in ovulation is much, much, much less. Now, to go into a bit of detail, for higher tier students, we need to understand in a bit more detail exactly what role hormones play in the menstrual cycle. Okay, so we've got a number of different hormones, and we'll sort of try and talk about them all together, but let's get an overview of them first. Hormone number one is estrogen. Estrogen is released by the ovaries, and its job is to thicken the uterus lining. Okay, so we can see here, this orange line is estrogen. It's known in America as estradiol, so that's why it's got a different word, different name here. And as the amount of estrogen goes up, you can see the uterus lining starts to thicken here. Okay. Now, FSH stands for follicle stimulating hormone, but just the initials FSH is fine. It's released by the pituitary, and it causes a follicle in the ovaries to mature an egg. Okay, and we can see that happening here. So this here shows the growth of a follicle over the first 13 days of the menstrual cycle, and inside the follicle is an egg, and that egg starts off immature and unready to be fertilized, but the job of the follicle is to fertilize it, so is to mature it rather. Okay. Now at around day 14, ovulation occurs, and you can see that happening here. The egg has been released from the follicle, and that is triggered by a hormone called LH, which stands for luteinizing hormone, but again, LH is fine. LH is also released by the pituitary, and it triggers ovulation. So we can see here, if we look at this graph, you see the blue line is LH. We get this great big spike of LH, and that is what triggers the ovulation. And then lastly, we've got progesterone. Now, the follicle here becomes a temporary organ and a temporary endocrine gland called the corpus luteum. And the corpus luteum produces the hormone progesterone. Now, progesterone does two things. Firstly, it maintains the uterus lining. So we can see here that all the while the amount of progesterone is high, the uterus lining stays nice and thick. Okay, but also it inhibits the release of follicle stimulating hormone, of FSH. So all the while that the corpus luteum is making progesterone, there is no FSH. So if we look here, while progesterone is high, FSH, the green line, is very low, and that means there are no more eggs being developed in the ovaries. Now, the thing with the corpus luteum is it is temporary. So if we look here, we see the corpus luteum gradually breaks down over the space of a couple of weeks, and eventually it stops producing progesterone, and that is what triggers menstruation. So we can see that as the amount of progesterone goes down, that is closely followed by the uterus lining breaking down in menstruation. But also, because there's no progesterone, there's nothing to inhibit the FSH, so that goes up again and starts to cause another follicle to develop. And so we begin the whole cycle again. Controlling blood glucose concentration. Now, the concentration of glucose in our blood is very important. We need glucose in the blood because glucose provides our cells with the fuel for respiration, which releases the energy we need. However, if the blood glucose concentration gets too high, it can cause serious and permanent damage to our cells and can lead to things like blindness and all sorts of stuff like that. But if the glucose concentration is too low, then we don't have enough fuel for respiration, and we can find ourselves in very serious trouble too. So the concentration of glucose in the blood is controlled by two hormones. We have insulin and glucagon. Now, both those hormones are released by the pancreas. Okay. Now, after we eat, the concentration of glucose in the blood increases because we digest the food and the glucose from the food is absorbed into the blood. This means that the concentration could become dangerously high. So the pancreas releases the hormone insulin, and insulin tells the liver to take the glucose and convert it into glycogen. Now, glycogen is a stored form of glucose, and this has the effect of reducing the glucose concentration in the blood. Between meals, as we walk and move and think and heat our bodies and do the daily business of life, we use up the glucose that is in the blood, and so the blood glucose concentration starts to decrease. Now, if it decreases too far, it could be dangerous for us. So now a second hormone from the pancreas steps in. This is called glucagon, and glucagon tells the liver to convert the glycogen back in to glucose. So glucagon has the effect of increasing the blood glucose concentration. So to summarize, after a meal, your blood glucose concentration increases and insulin is released to bring it down, converting glucose into glycogen. And between meals, as your blood glucose concentrations start to drop, glucagon is released, which has the effect of increasing blood glucose by telling the liver to convert glycogen back into glucose. Now, if you have type 1 diabetes, this is a disease that you are either born with or you develop during your lifetime as a result of your genes. It's to do with your immune system attacking the cells in your pancreas that make insulin. And what happens is you don't produce your own insulin, which means that when your blood glucose concentration increases after a meal, you don't have a way of easily bringing it back down. So people with type 1 diabetes, they must inject insulin in order to control their blood glucose concentrations after a meal. And if they don't get that right, they can create very serious health problems. People with type 2 diabetes also have trouble controlling their blood glucose concentrations after a meal, but this time it's not that the body doesn't produce insulin, it's either that it doesn't make enough insulin or that the body is making insulin but does not respond to it properly. And this is typically because a person has an unhealthy lifestyle or an unhealthy diet where they're eating so much sugar that their body can't produce enough insulin to remove it all from the blood, or that their body has produced so much insulin over its lifetime that their cells have stopped responding to it properly. They've become resistant to the insulin. And so type 2 diabetes is largely acquired during lifetime by an unhealthy lifestyle. That's one with a poor diet and too little exercise. So Not surprisingly, the main risk factor then for type 2 diabetes is obesity. So obesity we define as people with a body mass index of over 30, and body mass index is your mass in meters divided by your height squared in kilograms. We could also consider people with a high waist to hip ratio being at risk of type 2 diabetes as well. So that is people where the waist width divided by the hip width is an unhealthily high figure. Now, to treat type 2 diabetes, you can't just inject insulin, because the problem is the body doesn't respond to insulin well enough anyway, so simply injecting more insulin won't solve the problem. So instead there are a couple of things. One is to take medication that increases the sensitivity to insulin, so that the insulin that people's bodies are making has more effect than it was having. You can eat a less sugary diet and get more exercise, and it is these lifestyle factors that are going to be more important in controlling type 2 diabetes in the long term. Topic 8. Transport and exchange. So this looks at the idea of surface area to volume ratio, the circulatory system, respiration, and the core practical that was on this topic. Surface area is a really important idea in biology because it's to do with the exchange of substances. So if you think about getting oxygen from our from the lungs into our blood, or from our blood into our cells, that oxygen has to travel across a membrane from one cell into another cell. So the higher the surface area of those membranes, the more quickly that oxygen can be exchanged. And so this is an idea we'll see come up again and again in biology about maximising the surface area to allow substances to be exchanged more quickly. Some examples of that are the capillaries, the very fine blood vessels that run through all of our body, really big surface area to maximise how quickly substances can be exchanged between the blood and our cells. And also the alveoli in our lungs. So we've got some alveoli here. So these are the tiny little air sacs that in our lungs, and we've got millions of these little things, all with this really high surface area. You see how it's wrinkled like that to increase the surface area, and that maximises how quickly oxygen can be absorbed from the air into our blood, and how quickly carbon dioxide can be transferred from our blood back into the air in our lungs. Okay, and so we talk about this idea of surface area to volume ratio. Okay, and so the higher that ratio, the quicker substance exchange can take place. And to work out the ratio, you just do the surface area divided by the volume. It's dead easy. But as I said, a high surface area to volume ratio means that substance can be exchanged much more quickly. So the circulatory system is comprised of our blood vessels, and our heart, and they all work together to transport substances around the body. So they're going to carry oxygen and nutrients towards our cells, and they're going to carry carbon dioxide and other waste products like urea away from our cells. If we think about the blood vessels first of all, we've got three types of blood vessel: arteries, veins, and capillaries. Okay, now arteries carry blood away from the heart, and so because they're carrying blood away from the heart, they carry it under high pressure. So they need thick muscle walls. So if you look at this artery here, we see that the walls of that artery are physically thick. Okay, that is to give them a good deal of strength to withstand the high pressure of the blood that is pumped through them. And for the most part, they carry oxygenated blood. The only one that doesn't carry oxygenated blood is the pulmonary vein, carrying blood from the heart to the pulmonary artery rather, carrying blood from the heart to the lungs. Now veins, they carry blood towards the heart. Now that blood is under low pressure. So because of that, they've got these thin walls. If you look at the wall here, which is much thinner than the one on the artery. Okay. Now the blood is deoxygenated, with the exception of the pulmonary vein, which carries oxygenated blood from the lungs back to the heart, and they contain these valves. The valves make sure the blood can only flow one direction. So if we look here, that's a valve. Okay, so the blood can flow this direction through it. If the blood was to try and flow back in the other direction, that valve would be forced shut and the blood would not be able to flow. And then finally, we've got the capillaries. The capillaries are where the actual substance exchange takes place. So these are these very fine network of really super tiny blood vessels that are 1 or 2 red blood cells wide, and they connect the arteries to the veins, and that's where substance exchange happens. So here we have oxygen and nutrients leaving the blood, and carbon dioxide and waste products going from the cells back into the blood, and they have this really high surface area to volume ratio to make sure that that exchange of substances can take place as quickly as possible. Now the blood itself, we've got a few key components. We've got red blood cells, we've got white blood cells, platelets, and plasma. Red blood cells are also known as erythrocytes, and their job is to carry oxygen. So to help them carry oxygen more readily, they've got a biconcave shape. That is like a flattened disc with a curved indent on either side, and that increases the surface area so that they can absorb oxygen more quickly and release it more quickly. And they also have no nucleus. They're the only cells in the body that don't have a nucleus, and that just gives them more space to carry more hemoglobin. Remember, hemoglobin is the chemical in red blood cells that actually carries the oxygen. Now that's about 45% of the blood is the red blood cells. Roughly 1% of blood is white blood cells. Now, white blood cells, their job is to fight disease, and there are 2 types. We've got lymphocytes and phagocytes. Now, lymphocytes, they make antibodies. Okay, so you can see a lymphocyte here, and it's coated in these antibodies. Now, the job of antibodies is to stick to antigens on pathogens, so that's invading foreign cells, and that flags up to the immune system that they are sort of enemies to be destroyed. The job of phagocytes is to physically engulf the pathogens and then digest them, and you can see that happening here. Okay, so you see these little red dots represent bacteria, and you can see the way the phagocyte has just gulped it up. So it's just engulfing the bacteria. Eventually, the bacteria end up right inside the white blood cell, and then they'll be digested by that phagocyte. Also in blood we have platelets. Now platelets are tiny fragments of old cells whose job is to clot the blood. So if we have a cut or something like that, it's platelets that will help to start form the scab to stop our blood from just leaking away. And the last thing we got is plasma. Plasma is this straw-coloured liquid here, which makes up about 55% of our blood, and its job is twofold. Firstly, it actually carries the blood cells, and the second thing is it transports dissolved substances, so it can carry dissolved nutrients towards the cells and also dissolve waste products like carbon dioxide and urea away from the cells. Now the heart is a double pump, so it actually pumps on 2 circuits. Circuit number 1 is pumping the blood to the lungs and back. Okay, and the idea of that is to take deoxygenated blood to the lungs to get rid of its carbon dioxide and pick up oxygen and bring it back to the other side of the heart. Now it is the right hand side of the heart that pumps the blood to the lungs and back. And then the left hand side of the heart, it pumps the blood all the way around the rest of the body back to the right hand side of the heart. So the left side pumps the blood around the whole of the rest of the body, distributing the oxygen that it's picked up from the lungs as it does so. Now if we look at the structure of the heart, we've got these 2 chambers at the top. Okay, poor colour choice, I'll go for a better one. It's got these 2 chambers at the top. These are called atria or atriums. Okay, and then you've got these bigger chambers at the bottom here. Okay, and these are called ventricles. So blood enters the heart by the atrium, via the atria, and is pumped out of the heart by the ventricles. And the sequence of events goes something like this. So blood first enters the heart through the vena cava. The vena cava is the main vein coming from the body with all that deoxygenated blood, and it enters the right atrium of the heart. The right atrium contracts and forces the blood into the right ventricle. The right ventricle contracts and forces the blood out of the heart through the pulmonary artery. Now this blood is deoxygenated at the minute because it's come from the body and all the oxygen is being used up. Now the pulmonary artery carries the blood to the lungs, and in the lungs the blood loses carbon dioxide and it picks up oxygen, and it is carried back towards the heart in the pulmonary vein. The pulmonary vein is the only vein that carries oxygenated blood. From the pulmonary vein, it enters the heart by the left atrium. The left atrium contracts and forces blood into the left ventricle. The left ventricle contracts and forces the blood out of the heart through the aorta under really, really high pressure. And from the aorta, the blood branches off into smaller and smaller arteries until eventually it reaches the capillaries. And in the capillaries, the blood gives oxygen to the cells and removes carbon dioxide from the cells, and it makes its way into the veins. All the veins meet up to join up into the vena cava, and from the vena cava, the blood goes back into the right atrium again, and the whole cycle goes around once more. This whole process takes something like 2 minutes to complete one full lap. Okay. Now with the heart, we talk about the cardiac output. This is the total volume of blood pumped by the heart every minute, and it is the stroke volume times by the heart rate. The stroke volume is the volume of blood pumped by each single heartbeat, and the heart rate is the number of beats per minute. So the cardiac output is the two of those multiplied together. During exercise, both your stroke volume and your heart rate increase, so your cardiac output can increase massively. Respiration is the process of releasing energy from glucose, and there are two types of respiration. We've got aerobic and anaerobic. Aerobic respiration takes place in the mitochondria and involves oxygen. So the equation for that is glucose plus oxygen makes carbon dioxide and water. Okay. Now, during exercise, our demand for energy increases. So we're going to need to do more respiration. So to enable that, our heart rate increases and our breathing rate increases to get more oxygen into our blood to our cells. So more oxygen goes to the cells. That means the rate of respiration can increase and more energy can be released. So energy demand increases, the heart rate and breathing rate increase, which gets more oxygen to the cells, so there's more respiration, so more energy can be released. Now sometimes during exercise, aerobic respiration cannot provide enough energy, so we do anaerobic as well. Now anaerobic respiration takes place in the cytoplasm and has a much simpler equation, just glucose making lactic acid. It doesn't make much energy, but the energy it does make is made super quickly. So it gives us this little extra boost of energy during really intense exercise. You know, you've got to run away from a tiger, you're going to have a bit of a speed boost over your normal energy levels. However, the lactic acid that's made is poisonous. That means even if you only sprint for, say, a minute, 10 seconds or something, all the lactic acid that's built up has to be got rid of, and so you continue breathing really heavily for several minutes afterwards to make sure that lots of oxygen gets into your blood to oxidize that lactic acid to stop it from causing any harm. We did a core practical on respiration, and the aim of it was to investigate how temperature affects the rate of respiration of small animals, and with ours we used maggots. Now, the idea was we had a tube like this, okay, and in the bottom of that tube we put soda lime. Now, soda lime is a chemical that will absorb carbon dioxide, and that's a really important idea here. So we then put down a layer of gauze. That is just there to protect the animals we're using, because the soda lime can burn them because it's quite caustic. And we got 10 maggots and we put them in the tube. Then we placed a bung on the test tube, and it had a capillary tube attached to it like this. A capillary tube is just a really thin glass tube. Okay, and we placed it in a water bath for about 5 minutes just to make sure everything got up to the same temperature. Then what we did was we dabbed a little ink spot into the end of our capillary tube, and we started the timer. And the idea is that as those little maggots respired, they would use up oxygen from the air and breathe out carbon dioxide, but the carbon dioxide gets absorbed by the soda lime, so the total volume of air in the tube decreases, and that has the effect of sucking along the little dot of red ink. Okay, and so we can, after 5 minutes, we can measure how far the little spot of food colouring has travelled, and the further it's travelled, the faster the rate of respiration. And so we repeated this at a range of different temperatures, and we found that at a temperature of around 35 degrees Celsius, the ink spot moved the furthest because the rate of respiration was quickest. At the colder temperature, particularly in the ice bath, the ink dot moved a lot less far because respiration was much slower. Topic 9, ecosystems, in which we look at ecosystems, biotic and abiotic factors, measuring populations, and nutrient cycles. Ecosystems. An ecosystem is the organisms and the environment in which they live. So it is the two things combined together. And when we talk about ecosystems, we talk about a community. The community in an ecosystem is all of the organisms combined together of all different species. We can talk about populations. A population is the members of one individual species within an ecosystem. A habitat is a particular area within the ecosystem where certain organisms would live. So if your ecosystem was, say, a forest, a habitat within that forest might be a pond. Then we have food webs. Food webs like this, these show the feeding relationships in an ecosystem. So the arrows show what eats what, and the reason the arrow points from what is eaten to what eats it is because it shows where the energy goes. Now, if we look at our food webs, the base level is called the producers, okay, and that is almost always plants. Then we have the primary consumers, the secondary consumers, and so on. So we have these different levels within our food web. And right at the very top, we have the sort of the apex predators. Okay, and now you normally have a lot more things at the bottom and a lot fewer things at the top because the energy gets less and less the further up you go. Now, if we want to measure a population, we can use random sampling to do that. So random sampling involves estimating the population of something by dropping quadrats at random. So for example, in this field here, if we wanted to estimate the number of sunflowers, we could drop a quadrat. Here's my little quadrat, okay? Now you drop them at random, you're not aiming for a particular sunflower, you just drop it at random, and the idea is you count how many sunflowers you can see in that quadrat, okay? Do that, I know, 10 times and take an average. And then to work out or to estimate the overall population, you just multiply the number of organisms in one quadrat by the total area divided by the area of the quadrat. This term here just scales up one quadrat to the whole area that you're investigating. And this will give you an estimate of how many, of the size of the population of the organism that you're looking at. We did a core practical using quadrats, but it wasn't to estimate the total population. It was to investigate this question, which was, how does the number of daisies or some other plant vary as you move away from a tree? And to do that, we use a method called a belt transect. So what we did was we placed a quadrat at the base of a tree, okay, and we counted and recorded the number of daisies. Then we moved it a metre away, and we repeated, counted the number of daisies, and moved it another metre away, and so on, all the way up until you're about 20 metres away from the tree. And then you repeat this with 2 more trees, so you can take an average. And what we found was that the number of daisies started off low and then increased as you moved away from the tree, but then it levelled out. And the reason why it increases is because near the tree, you know, the tree is using up all the nutrients, the water in the soil is also blocking a lot of the light. So the daisies don't have everything that they need, but the further away you go, the less effect the tree has, and so the more freely the daisies can grow. Now, in deciding what lives where, in because different species live in different places, we consider both the abiotic factors and the biotic factors. So this affects what lives where. So abiotic factors are the non-living influences on what lives in a given place. So we can talk about the climate, so the temperature, the rainfall, that has a big effect. The type of soil, the materials it's made of, the pH of the soil and so on, and pollution is an example of a man-made abiotic factor. So all of these things will affect what can live where. Biotic factors are living things that affect what can live where. So, most animals, for example, need food. So what plants are available for food? What plants are available for shelter? And what predators might there be that might want to eat you? What prey is around that you might want to eat? And what diseases exist in that area? So these are all living factors that affect what can live in a given place. And one sort of related example of this is the idea of predator-prey cycles. So predator-prey cycles describe how the numbers of predators and prey animals change over time. So if we start with, we've got rabbits and wolves here. So if we start with assume the number of wolves is low, the number of rabbits increases because there's not much eating them. Okay. However, as the number of rabbits increases, the number of wolves starts to increase too because the wolves have more food. But eventually the number of wolves gets so high that they overeat the rabbits and the number of rabbits goes down. With less food around, the number of wolves goes down. Okay. With less wolves around, there's less to eat the rabbits, so the number of rabbits starts to increase again. With more rabbits around, the number of wolves starts to increase because there's more food. But again, then the number of wolves gets too high and they overeat the rabbits and the number of rabbits comes down. And so you get these cycles of increases and decreases over time as the numbers of rabbits and wolves adjust to each other. Parasitism and mutualism is a couple of really interesting examples of the way that organisms can interact with each other. So parasitism is the idea that one organism can feed off a host organism without benefiting it. So think about the way that leeches, here's a leech, can suck your blood. The leech gets a meal, we get nothing. Fleas do similar, suck our blood, we get nothing, they get a meal. Tapeworms live inside our digestive systems. They get a free meal, we get nothing from them. Up here on this picture is mistletoe, these big green bunches of leaves that don't look like they're part of the main tree. They put their roots into the plant and they rob it of its sap, but they don't give anything back to the tree. That's parasitism. The alternative, I think a better alternative, it always seems nicer to me, is mutualism. So mutualism is when two organisms work together to their mutual benefit. They both benefit from this. A good example you can see here. These little fish swimming inside the shark's mouth, they're called cleaner wrasse or cleaner fish. And they will go into the shark's mouth and clear bits of rotting meat or clear any parasites and keep the shark's mouth in good condition so it stays healthy. Now the shark will not eat those fish because the fish are providing it a service. Both animals benefit in this case. It is not in the shark's interest to eat the fish because the fish give it a better service by cleaning it. Another good example is coral and algae. You've seen coral reefs, they're made of these tiny animals called polyps, and algae live inside the polyps. Algae get shelter from the coral, and in return they provide the coral with some sugars and food from photosynthesis. So again, both organisms benefit from the relationship. Now humans, sadly, have had a pretty major impact on biodiversity. Biodiversity is the number of different species living in an area, and high biodiversity is an unquestionably good thing. In areas with high biodiversity, they are more resilient to changes and to disasters and to problems because there's a wider range of animals to absorb the impact. And also there are lots of undiscovered species that may benefit humanity, so high biodiversity is a good thing. But we've had lots of negative impacts on it. One example is introduced species. This is when, sometimes on purpose, sometimes by accident, we move species from one part of the world to another, to a part of the world where they've got no natural predators. A good example of this is cane toads, which were introduced to Australia to help control pests. But because cane toads have no natural predators in Australia, they've run out of control and they're now causing massive damage to native wildlife. Fish farming is something we do to produce fish in a large scale without having to go fishing. So the fish are grown in these great big nets that are, or net pens that are moored out at the sea. Produces lots of fish, but the trouble is that all the waste from those fish causes massive pollution to the surrounding area and really decreases biodiversity. And then up here we've got eutrophication. Sorry for the crossing arrows there. We've got eutrophication. So the idea here is that fertilizer from farmers' fields gets washed off the fields by rain and it makes its way into the lakes. And in the lakes it causes algae to grow really quickly. And that blocks the light out from the bottom of the lake. So anything living on the bottom of the lake dies because it gets no sunlight. And it decomposes. And bacteria use up all the oxygen while they're decomposing. And so that means all the fish and things that breathe the oxygen that's in the water, they die, they suffocate because there's a lack of oxygen. And so the whole ecosystem in the lake can collapse because the fertilizer causes this massive growth of algae. Now, we're not all bad. We are trying to overcome some of the problems we created, and that's called conservation. So conservation is the efforts of humans to improve or sort of restore damaged natural environments. And there's a few things we do. One idea is reforestation. So if we plant forests that we've chopped down, that massively increases the biodiversity of an area. And also we have captive breeding programs. So we use this for things like, tigers that are almost extinct in the wild because of hunting. So captive breeding means you can breed them in zoos and things and then release them back into the wild to increase the wild numbers. The water cycle describes how water is continuously being cycled between different sort of sources and stores within the environment. So it doesn't matter where you start because it's a cycle, but we'll start with precipitation. So precipitation is rain, but also snow and hail as well. And so this is when water comes down from clouds in the sky and hits the land. Now when it's on the land, collection happens. That is the way that water, gets soaked into the soil and it runs into streams or it falls into rivers or whatever, and the water gets collected into lakes and the oceans. Now from lakes, but mostly from the oceans, water is evaporated. So it turns into water vapour and goes back up into the air. Once it gets high enough, it condenses to form clouds. Okay. Eventually those clouds get so heavy and full of large droplets that precipitation happens, and the whole cycle goes round again. Now, humans need to drink water, so potable water is drinking water, and in order to treat water, we can't really just drink water straight from rivers and lakes and things, so we filter it first to remove dirt and floating debris and little bits of suspended gunk and stuff. And also we then treat it with chemicals to kill any pathogens. And the main chemical we treat it with is chlorine, which is what gives water a funny taste. We can also, in very hot countries, if there's not enough fresh water, we can distill seawater. So we heat it and collect the water vapour and condense it back to liquid, and that can produce water that way, but it's very expensive to do so. The carbon cycle describes how carbon is moved between different stores in the environment and the earth. So it doesn't matter where we start, but if we start with carbon in the form of carbon dioxide in the air, okay. Now plants photosynthesize, and so the carbon will go into the plant from the air. Okay. Now from there, plants do respire, and that will turn some of the carbon straight back into carbon dioxide. Okay. However, most of it doesn't. That doesn't happen to most of it. The main thing happens is plants get eaten by animals. Okay. And so the carbon is transferred into the animals, and some animals eat other animals and whatever. Okay. And those animals will breathe some of that carbon back out as carbon dioxide back into the air. Okay. And some of it will come out of them either in the form of waste, feces, poo, okay, but also in the form of their dead bodies, and it will become part of, it will fall to the ground. And in the ground, a few things can happen. Firstly, their bodies can be decomposed by, microorganisms and fungi and things like that. And as those fungi respire, they can put the carbon dioxide back, or carbon back into the air as carbon dioxide. Some of it, though, never gets decomposed, and it ends up eventually becoming fossil fuels like coal and oil and natural gas. Now, if we had any luck, it would stay there, but unfortunately we keep on burning this stuff. So burning coal and oil and natural gas is sending that carbon back into the air as carbon dioxide. Now we should also recognise that the oceans have a role here as well. So carbon dioxide is easily dissolved from the air into the water, and can easily leave the water and go back into the air as well. And exactly what takes place depends on a range of different factors. And finally, the nitrogen cycle. Nitrogen is super important because nitrogen is the basis of making proteins. Okay. Now proteins do every important job in the body, so without nitrogen, without proteins, we can't really have life as we know it. Now the problem is that nitrogen in the form of nitrogen gas in the air is really unreactive, so we can't really just use it. We can't breathe it in and use the nitrogen from the air. So instead, nitrogen has to be fixed, and it gets fixed in two ways. Okay, one is by lightning, like that, and the other one is by nitrogen fixing bacteria. Either way, it doesn't really matter too much which, but the point is that these bacteria will convert nitrogen into this form, nitrate here. Okay. Now nitrate is a mineral ion that can be absorbed by plants, okay, so it goes into the plants through their roots, and they can use it to make amino acids, and from the amino acids they can make proteins. Now once it's in the plants, it's very straightforward. The plants get eaten by the animals, and that transfers the nitrogen into them. The animals get eaten by other animals, and so on, transferring nitrogen from one animal to another. Now the animals are going to excrete, so urinate and defecate and produce waste, and that waste will contain some nitrogen, and that waste will be broken down by bacteria, first into ammonia here, and then it'll be converted by nitrifying bacteria back into nitrates to be recycled again by other plants. Or it could be that the ammonia gets attacked by denitrifying so the nitrates rather get used by denitrifying bacteria, which are really unhelpful. They convert it back into nitrogen gas back in the air. So that is the nitrogen cycle. Well done for getting this far. You have reached the end of this presentation. As I said right at the very beginning, make sure that listening isn't all you're doing. You must do stuff that makes you think, because when you think, you learn. Thank you, and goodbye.